So the next column chromatography technique is size exclusion chromatography. Size exclusion chromatography. So protein is separated on the basis of their size through size exclusion chromatography. So in case of size exclusion chromatography, the column, so this is the column, and the column is filled with beads, and in this case the beads contain cavities and pores. So the small proteins enter into the cavities and they have to pass through the labyrinthine, or you can say twisted path inside the beads. So slow proteins takes more time. On the other hand, larger protein molecules easily elute out because larger protein does not able to enter into the cavities through the pore inside the beads. So larger proteins take short path and elute earlier. So through this process we can easily separate the large proteins and small proteins. So these are the beads are arranged in this way, and these beads contain cavities and pores. So now the solution, on the other hand, the solution contain, so this is the solution contain small proteins and large protein. So these are some small proteins and a large protein present inside the solution. So at first the solution is present on the upper side of the column and moves inside the column, the matrix. So the buffer is used, and so this is the larger molecules, or you can say larger proteins elute earlier because they takes less time to pass through the matrix. This is the medium size the proteins. On the other hand, the smaller proteins remains on the upper side of the column, as you can see, because small proteins enter into the cavities and they have to pass through the twisted path inside the beads. And here it is the buffer reservoir, content buffer. So this is the buffer solution. So in case of size exclusion chromatography, proteins are separated on the basis of their size, and we can easily collect the large and small protein in tubes. So these are the tubes, and the large we can collect large and small protein in those tubes. So size exclusion chromatography is also very important process. In case of ion exchange chromatography, the proteins are separated on the basis of their charge. On the other hand, in case of size exclusion chromatography, they are separated on the basis of their size. So in case of size exclusion chromatography, the column is filled with cross-linked column is filled with cross-linked polymer. Column is filled with cross-linked polymer beads. Beads are beads are engineered. Beads are engineered with pores. So beads are engineered with pores. So these are beads. So the beads are engineered with pores. So as you can see from this diagram, these are the beads. So magnification of the beads look like that. So these are the beads and cross-linked beads, and inside the beads, so the smaller size protein enters inside the bead through the pores. So that is why the movement of the smaller proteins are retarded by these cavities or twisted paths. So these are the twisted path inside the bead. So bead content, so the molecules have to pass through the labyrinthine path through the bead, so it takes more time. So small protein takes more times. On the other hand, large protein does not enter inside the bead and elute earlier. So these are the large proteins. Large protein, the size of the pore is so less than the large protein, so large protein does not enter inside the bead. Now small protein small protein enter into the pores of the bead. So small proteins enter into the pores, and the movement slow down, and the movement of the small protein slow down because of the twisted path inside the cavities, because of twisted path through the bead. So cross-linked polymer, column contain cross-linked polymers beads. Column is filled with cross-linked polymer beads. Now beads, so the movement slow down. Small protein enters into the pore and their movement slow down because of twisted path through the bead. So because of twisted path through the bead. On the other side, large protein large protein does not large protein does not enter into the beads. Large protein does not enter into the bead due to their large size. So large protein does not enter into the bead due to their large size and moves faster and elute earlier. So large protein does not enter into the bead due to their large size, moves faster and earlier. Elute earlier. So this is the